Our next speaker is Taeok, professor from the Department of Microbiology and Immunology at the IUN School of Medicine, Northwest. And the title is Fighting Back Against Superbugs, a Novel Approach to Reviving Inactive Antibiotics.
Thank you. Hi, I'm Ted Beck, and I'm teaching medical microbiology at the medical school. And today, I'll talk about a new approach to fight back against the antibiotic-resistant bacteria, so-called superbugs. So the era of antibiotics began when Sir Alexander Fleming discovered penicillin in 1928, and he actually contaminated his bacterial plate, with a fungus called penicillium. And he noticed that around that penicillium, bacteria didn't grow. So he thought that something from that fungus killed bacteria. And that something turned out to be a penicillin. Penicillin kills bacteria by inhibiting cell wall synthesis enzyme called penicillin binding protein and blocking cell wall synthesis. And cell wall is very important for the life of bacteria. And then if you block the cell wall synthesis with penicillin, bacteria basically explode like that. Next. Antibiotics actually contributed in many parts to that dramatic decrease of the human death rate in early 20th century. But as you know that these days antibiotic resistance is a big problem. It is a global problem. And according to a report, by 2050, each year about 10 million people will die because of this problem. And that number is very similar to the number of people die from cancer. The reason we have this problem is it's getting harder and harder to develop new class of antibiotics, as you can see in this graph. These are 6 important antibiotic resistant bacteria, and today I'll talk about this methicillin resistant Staphylococcus aureus, aka MRSA. Staphylococcus aureus is a round-shaped bacteria, as you can see here. About 30% of people have this bacterium in their nose and on their skin. Usually this bacterium causes skin disease like this, but once they invade human body, they can cause infection in any body part, like blood, heart, lung, bone, joint. And then also it produces a toxin which can cause shock and kill people. MRSA is resistant to almost all penicillin class antibiotics, also resistant to many others such as aminoglycoside, macrolide, and tetracyclines. So if we cannot develop new antibiotics fast enough, how about reviving these ineffective antibiotics? Can you do that? And we and other labs think, yes, we can, by using this interdependent nature of the life. So in life, we depend on each other, right? And in MRSA, the resistance to the penicillin class antibiotics depends on the presence of a molecule called lipoteichoic acid, for short LTA. LTA is a polymer of phosphoglycerol, and it is attached to the cell membrane. If we remove this LTA from MRSA becomes very sensitive to penicillin class antibiotics. In this picture, the white strip is coated with oxacillin, a penicillin class antibiotics, from high concentration to low concentration. With LTA, MRSA can be killed only at high concentration of drug. But without LTA, it can be killed in low concentration of drug. In animal experiment, without LTA, MRSA can kill mice efficiently. But if we treat that infected mice with oxacillin, with LTA, MRSA still kill mice, but without LTA, MRSA cannot. So this is a proof of concept that by inhibiting LTA synthesis of MRSA, we can transform this formidable foe into an enemy we can handle and manage. Imagine if we develop a small molecule that can inhibit LTA synthesis in MRSA. By combining this small molecule to this or ineffective penicillin class antibiotics, we can revive their bacterial killing activity. And then that combined drug can be used to treat MRSA infection. I don't think this approach will solve all the antibiotic resistant problem at once, but I'm sure that it will reduce the burden of the problem significantly. And this is the new approach my lab and other labs are taking to tackle the problem. Thank you very much.